So we were talking about the genetic diversity, and let's talk about the genetic diversity. I'm going to, it's mentioned over here, that's the genetic diversity. Genetic diversity. Now, let's suppose we have a species A, and species A has a total number of 12 genes in the genome or in the gene pool. So species A has 12 genes. Maybe they have, we are not talking about the chromosomes. Might there be 6 chromosomes, 5 chromosomes, 3 chromosomes. We are talking about the number of genes, total number of genes. Like imagine as a human being, I told you, suppose we have chromosome number 1. And the chromosome number when we have 50 genes. This is for black eyes, I mean this is for the eye, for the height, for the texture of hair, for skin color, for insulin, for CFTR protein. We have different genes and they are located on one chromosomes. In the other chromosome there should be other genes. So if we count all these genes, this is called a gene pool, and this gene pool is similar in all the human being. So here 12 genes in the genome in species A. Similarly, we have species B, which has, let's suppose, 10 genes in the genome. Species C has 20 genes in the genomes, and species D has, suppose, that should be like 15, okay, it should be 18 genes in the genome. Now, if this is one community, I mean this is community 1. In this community we have 4 species, so the species richness is once again 4 here, and these number cannot be added as they are not representing the they are not representing basically the abundance, because abundance is for the number of individuals. These are not the number of individuals, these are the number of genes in one species. So 12 plus 10, it's 22, plus 20, that's 48, and then, it's 22 plus 20, so that should be 42 plus 18 means 60. So 60 different genes we have. 60 different genes we have in community 1. Now, suppose if there is community, there's another community. Oops, I have to rub this one. So, like here. Now, if we have, there is another community here. This is community 2. Here we have species S, species T, species U, and species V. Of course, they are different species, so they have different number of genes. And if S has, let's suppose it has 15 genes, T has only 8 genes, U has, it should be like 17 genes. So it may be like 15 plus 8, 23, plus 17 is 40, and here it has 15 genes. So the total number of genes here, we have like 15 plus 23, 40, and then plus 15, so 55. 55 different genes in community 2 than in community 1. In this connection, we say that genetic diversity of community 1 is greater than the community 2, but we are not sure still, because we cannot decide only on the number of genes. There's another factor as well. Like in species diversity, we were dependent on 2 factors, richness and abundance. Here we also dependent on 2 factors. Total number of different genes in a community, which is like here, total number of different genes from each species, different genes, we have 60, and here 55. This is one factor. And the other factor, how many alleles each gene possess, is also to be account. Because like, I'm just giving you one example of the blood group. So blood group, there's only one gene for the blood group, but they have three different alleles. I mean, blood group A, blood group B, or blood group O. So we have three alleles for the blood group. Similarly, for those characters which are continuous character, like for this color of skin, or you can say like the height of an individual, body mass. Now these are some continuous characters, and we have different genes and even different alleles as well. So if there's a greater number of alleles per gene, then we should also take into account the number of alleles as well. And now, giving you an example, same here. Suppose in these 60 genes, there are here, I'm just like from here, these 60 genes, which is the total number of genes in community 1, and in these 60, 25 genes have 4 alleles per gene. So we just have 25, one gene has 4 different alleles, and we have 25 genes. So if you multiply by 4, it means we have 100 different phenotypic expressions. I mean alleles, because allele is responsible for the phenotypic expression and gene is responsible for a character. You know, we have discussed in detail about these two, because character is character and expression is expression. Again, I'm giving example like we say blood group, so it's a character. Expression A, B, or O. Height is a character. Tall, dwarf, that is the expression, I mean phenotypic expression. Color of eye is a gene. Blue, black, brown, these are expression. So expression is represented by alleles. So we have 100 different types of expressions for 25 gene. And the rest of the gene, which are the remaining genes, are how much? They should be 35. So the 35 gene, each of them have just only 2 alleles. Let's suppose 2 alleles. So if there are 2 alleles, we have 35 multiplied by 2. So we have 70 different types of allele. So we combine these 2 alleles, the total gene pool, along in the sense of alleles as well as gene, we have 170 different phenotypic potential. I mean, this community have the potential to produce 170 different types of characteristics features. So it has, this is the potential of the diversity in the sense of genes. Now look this one. Here we have total number of gene 55, but let's suppose in these 55, total of 55, we say that let's about the 20 genes. Each of the 20 genes possess about 6 alleles. 6 alleles. So we just have to multiply by 6. And the remaining, how much? The remaining 35, each of them have 3 alleles. And just like keep in mind, this is just an example. Like maybe most of them have 2 alleles, but like an example. So we just have 35 multiplied by 3. These are the number of alleles. And so here we have 120 phenotypic expressions. And here we have 3 5s are 15, carry 1, 3 3s are 9, 1 10. So 105 expression. So the combined allelic potential should be here as it's 2, 2, 5. It's 225 is the phenotypic expressions pool. So that's why I'm saying